Greetings, Brookings APBIO students. This is Mrs. Rydell, and this is the continuation of our DNA technology slideshow. You should have watched the beginning part when we looked at plasmids a little bit and how they can be used for making recombinant organisms and recombinant pieces of DNA. We looked at engineering plasmids, creating plasmids that have combinations of genes from two different organisms. Usually plasmids are carrying genes for antibiotic resistance. We can take advantage of that by using that property to select for those plasmids that contain the recombinant DNA. We talked about restriction sites and cutting the DNA using restriction enzymes. And now we're going to look a little bit at how we can analyze that DNA, look at DNA between different organisms. That has to do with DNA fingerprinting. So if you think about those restriction enzymes now that we know we can cut DNA, use those molecular scissors to cut the DNA in a variety of ways, we can now compare DNA between organisms for a variety of reasons. For example, in crime scene forensic analysis, comparing suspects to crime scene evidence, looking at medical diagnostics, comparing genes between people who have genetic disorders and those who don't. We can use that as a diagnostic tool to look for carriers, to look for people who have certain mutations that can cause them genetic problems. Used for paternity testing, who's the dad? We can use DNA fingerprinting to identify parents and children. Evolutionary relationships, we use it to study evolutionary patterns and look for relatedness between organisms. And there's a whole variety of reasons to use DNA. Example would be looking at endangered species and collecting evidence against poachers. How do we compare the DNA fragments now? We're going to cut the DNA using those restriction enzymes, and then we're going to separate the fragments based on size. We're going to use a gelatin base, usually agarose, which is kind of like the same thickener that comes from an algae. It makes kind of like a thick jello, what we call a matrix. And then we're going to do a process called gel electrophoresis. We're going to add electricity to the gel, and that's going to cause the little fragments of DNA to move. If you remember the structure of DNA, DNA has phosphate sugar background that makes it negatively charged. So if you add an electric current to the gel, we're going to make a negative on one side and positive on the other end. And we add DNA to the gel by placing it in a little well. And then applying the current, the fragments of DNA will move along the gel, and they're going to move toward the positive pole on the gel. Kind of like swimming through jello. If you think of a big swimming pool full of jello, and you trying to move from one end of the pool to the other. The fragments now, as they move down the gel, are going to be separated by their size. The size of the fragment determines how far it's going to move in the gel. Smaller pieces are going to travel farther than bigger pieces. So they're quicker, they're faster. Think about if you had a race between a 400-pound sumo wrestler and a little wiry track kid. The smaller pieces can move faster. Sometimes I like to think of it as instead of swimming through jello, running through a cornfield. And if you think about how fast you could get from one side of the cornfield to the other, if you were running kind of in between and weaving in and around the stalks, that's what the fragments are doing. They're moving through this jello matrix. And the smaller pieces are going to be able to get through the matrix faster. Think about running through a cornfield with one person, a race versus people, maybe a group holding hands and making a chain and trying to move through that jello. So this is how we're going to set up our process. We're going to have a frame that holds the jello, holds the agarose gel, and we're going to treat our DNA with restriction enzymes to cut it into pieces, and then we're going to add a power source to the gel and move the fragments along the gel and then look for patterns. We're going to stain the gel to see where the pieces of DNA end up, and you can compare different samples of DNA and see what kind of banding pattern is produced on the gel. Longer fragments are going to move shorter distance, smaller fragments are going to move a farther distance on the gel. You look at running the gel and then staining. Okay? One of the stains that we can use is called ethidium bromide, and it actually is a stain that binds to DNA, has an attraction for the DNA, and it glows under UV light. So if you look at this little gel picture here, you can see how it's going to glow or fluoresce. This has been stained with something called ethidium bromide. Ethidium bromide is a serious carcinogen, serious mutagen. Because it binds to DNA, obviously we don't want to be using that chemical in the classroom. And so we are going to use another stain called bromethymol blue that will is less carcinogenic and recommended for our high school kids to use. So we are going to stain ours a little bit differently than the fluorescent stain, but we're going to stain our gel so we can see the bands. Remember, DNA is tiny. It's a molecule. You're not going to be able to see it with the naked eye. And we're going to kind of set up a little crime scene in our lab that we're going to do, and analyze some DNA from different individuals and compare them. So you'll see a gel pattern and be able to compare sequences between different organisms. Example of how we might be able to use this. We can use DNA analysis to compare DNA from different organisms, for example, and look at evolutionary relationships. Which patterns have more in common with each other? And if you look at the patterns here, you can see 1 and 2 have a very similar pattern, except for this band here. 3 and 4 have very similar pattern, except for this one band. And so we can compare DNA between different organisms and create kind of a little family tree by looking at similarities between DNA. We're going to do a little lab, also a blast lab, that looks at DNA sequences and compares similarities between organisms. We can use this for diagnostic purposes. One of the first evidences that was found for organisms who had genetic mutation was comparing hemoglobin. You can run proteins on gels in addition to DNA, comparing hemoglobin between a person who had sickle cell disease and a person without that mutation. And that was done. Linus Pauling was the person who did that experiment and showed that there was actually a change in the protein molecule when it was run on a gel. So comparing, because these sequences are going to be different. If you have a mutation, they're going to make different gel patterns. You can use it as a test for carriers. You can use it as a test for to determine if someone has a certain mutation by running their DNA on a gel. We use it oftentimes for crime scenes, comparing DNA from different suspects with a crime scene, maybe blood or semen that's collected at a crime scene, and comparing to determine the suspect, who is the guilty person. If you compare this, obviously if this is my crime scene data that came from the crime scene, and here are my 3 suspects, you can see who's a match for the crime scene data. Comparing blood samples on clothing, we can compare semen samples collected in, for example, rape cases, and then looking at the banding on the gel to see who's the guilty person. So in this case, we're comparing defendants, the defendant's blood, the suspect's blood, and victim's blood. And here you can see the victim's blood is on the suspect's shirt. Collected sample evidence and looking for those patterns. And that it kind of creates what we call a fingerprint. Almost like looking at fingerprints on a left behind when you touch something. This pattern is kind of what we call a fingerprint, and we can compare fingerprints between different. Everyone has a unique pattern because of everyone's DNA is a little bit different. So what makes those differences? If you look at the patterns that we see in those gels, it comes from differences in the DNA sequence. And the differences probably aren't in the actual gene patterns, the gene code. Everyone is a little bit different as far as like hair color or eye color, but the big differences are going to come in those areas that we call junk DNA, the areas that don't really code for proteins, kind of those in-between patterns. And everyone has repeats, different kinds of patterns in that junk DNA that allows us to separate and compare sequences. If you think about the places on your DNA, 23 different chromosomes, think of all the possibilities for those little bit of difference. So if this is an area, a non-coding area, it's not going to affect the person's phenotype, how they look, what color eyes they have, but they may be a little bit different than another person. And often these repeats, areas where we have repeats over and over again, are where we're going to be able to see differences between organisms. So if you look at a pattern, here's an example of a piece of DNA, and remember our restriction sites now are going to cut the DNA into fragments, and we're going to use restriction enzymes to do that. And if I run those pieces on a gel now, take my cut DNA, I'm going to have 3 fragments, and I'm going to take the little gel and add my DNA, and if I run it on a gel now, I'm going to see a band based on the sizes of the fragments that are cut. So the biggest piece, I'm going to take the slowest time getting down the gel, and based on size now, you can see the different fragments where they end up on the gel. Comparing between people, if you look at this section here now, this repeat area, if I compare the DNA from another person that has more repeats, their fragments are going to be a little different sizes. These restriction sites are going to cut in the same place, but this center fragment now is going to be a little bit bigger because there are more repeats here. And so when I run this on the gel, I'm going to see a little bigger piece here, which is going to not move quite as far as this piece. And you're going to see these 2 pieces, those stay the same, and you're going to see a different banding pattern based on those differences between those, what we call the junk DNA. Those differences are called restriction fragment length polymorphisms. Many different shapes, and we're going to get some different sizes of fragments based on those differences between individuals. So when we run their DNA, right, changing the sequence affects where the restriction enzymes cut, changes the sizes of the pieces you get when you make cuts, and it's going to allow us to make a fingerprint, create different band sizes on the gels when we compare individuals. This technique of DNA fingerprinting was invented or just created by a guy named Alec Jeffreys in England back in 1984. And restriction fragment length polymorphism analysis, RFLP analysis, is the official name for what we call DNA fingerprinting. We're analyzing those differences between individuals.
Restriction enzymes recognize very specific sequences of nucleotides in DNA. DNAs from different individuals rarely have exactly the same array of restriction sites and distances between these sites. Therefore, the population is said to be polymorphic, having many forms for these restriction fragment patterns. These differences are referred to as restriction fragment length polymorphisms, RFLPs. Such differences may arise through mutations. By cutting a DNA sample with a particular restriction enzyme, DNA fragments of different length are obtained. These fragments are separated by gel electrophoresis. This provides a pattern of bands that is unique for the particular DNA being analyzed. These DNA fingerprints are used in forensic science during criminal investigations. RFLPs are also useful as markers to identify particular groups of people at risk for certain genetic disorders.
One of the ways that RFLP analysis can be used is for evidence in trials, looking at crime scene evidence. And the first case was used successfully in 1987 to convict a rapist and using DNA evidence. You can see the sample taken from the rapist and blood sample taken from the suspect, comparing against the crime scene data versus your suspect data, and you can see similarities between the patterns on the gel. We always run standards, and we're gonna, when we run our DNA, we're gonna run we call it a ladder down the side that runs DNA of known sizes, so you can have a little size comparison to compare against. This is another sample taken. You can see the sample from the crime scene sample and comparing against the suspect. So how do we compare DNA from blood or semen, for example? Remember, these are cells from the same person, whether they're coming from reproductive or whether they're coming from blood, they're going to contain the same kinds of DNA, so you could compare blood versus semen. Issue comes with red blood cells. Remember, when we're taking blood samples, red blood cells do not contain DNA in mammals. In humans, they lose their nucleus so that they have more room to carry oxygen. But there are other cells in blood, white blood cells, for example, that do have their nucleus intact. And so when we're doing blood analysis, we're looking at the DNA taken from white blood cells. This is evidence in a murder trial, and you can look to see, here is blood samples from the crime scene, obviously blood from multiple people at the crime scene, and here's blood samples. When there's a crime, we always take blood from the victim and suspect so we can compare against and look to see. Now here is blood from victim number 2. This is blood from victim number 1. You can see this blood at the crime scene came from this person. If you look now, this is my blood from my suspect, and look to see, here's suspect blood from the crime scene. Obviously the suspect left some blood behind. This is an actual evidence from an actual crime. If you remember, a few years ago, O.J. Simpson, the football player, was accused of murdering his wife and a friend of hers. And you can see this is blood from the suspect, O.J., found at the crime scene. And this is his wife, her blood also at the crime scene. And it suggests he's the guy. Using DNA as a way to determine paternity, comparing parents and kids. If this is the mom, and here are the two possible dads, this is the child's DNA. If you have a child produced by parents, right, that child is going to have fragments, DNA sequences inherited half from mom and half from dad. So if you look at the banding from the child's lane, you can tell where those sequences came from. In this case, all the people have this band. But if you look here now, if you see a band in the child's lane, it either has to come from mom or come from dad. In this case, here's a band that the child has that shares in common with this dad. Here's a band in the child's lane that came from mom. Here's a band that came from dad. Here's a band that came from dad. This one came from mom. And if you compare, notice the child doesn't have all the bands that this dad has, but every band in this lane is either going to match up with a band in the dad lane or a band in the mom lane. You can tell by looking at this pattern. Here's the mom of this child, and this guy, person number 2, is gotta be the dad. You can tell by comparing sequences between parents, what the child has compared to the parents. One of the things we did before was when we put our plasmid, our recombinant plasmid, into our bacteria and then allow the bacteria to reproduce to make lots and lots of bacteria containing that plasmid, that takes some time. And so what we have since come up with is an easier way of making lots and lots of copies of DNA using another technique, and allows us to do it without growing it in bacteria cells. And that's something called PCR, polymerase chain reaction. We can now copy DNA, very small amounts of DNA, for example, collected at a crime scene, maybe a little drop of blood, and use a machine rather than the bacteria to make multiple copies. Get enough DNA to run on a gel, for example. We only need one cell to get the DNA collected from that one cell, and then use the PCR machine to make multiple copies. We copy the copies, make copies, we make copies of the copies, and in a very short amount of time we can have huge copies of a piece of DNA that allows us to do analysis in electrophoresis. So we're going to do the copies artificially in a test tube. So what do we need to do this? We need a template strand that's going to be collected at the crime scene, for example. We need DNA polymerase that's going to make the DNA copies. We need nucleotide subunits to add together to make do the replication. And we're going to use the ATP, GTP, CTP, remember, built-in energy supplied by those phosphate groups. And we need something called primers. Remember, DNA polymerase can't start a chain by itself. It needs a primer to get it going. And so we're going to start with our little strand of DNA. And primers now are going to be the key. We need a primer that's going to attach to the DNA, and it has to be a match. So we have to have primers that match the little piece of DNA that we're going to start sequencing. And then we're going to put in our primers, and make the copies. Now, primers are going to kind of, whatever primers used are going to kind of determine the section of DNA that you're going to make the copy of.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA, and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
So we're going to use this process. We're going to put our little piece of DNA into a machine called a thermocycler, and we're going to do PCR, polymerase chain reaction. We're using polymerase to make copies where heat is going to separate our strands. Polymerase is going to fill in the matches, and we run the cycler over, and it's a cycle over and over and over again to make multiple copies. So think about this process now. We're going to heat the DNA up to about 90 degrees. So it gets very hot. That separates the strands. And we're going to repeat the cycle of denaturing and then annealing, hybridizing, putting the primers in and building the DNA strand, and then heat again to separate those strands. And so if you think about it now, think about what you know about enzymes and heat. What is going to happen to our DNA polymerase if we heat it up to 90 degrees centigrade? That's almost to boiling temperature. Remember, 100 degrees is boiling, so it gets very hot. And if you remember what happens to enzymes when you heat them, they denature, they stop working. So in order to run this process, we need an enzyme that's going to be heat stable, what we call heat stable, not denatured or unwound when we heat it up. And so the way this works is scientists have come up with a form of DNA polymerase that can stand high temperatures. Where do we get it from? It's called Taq polymerase, and it comes from a bacteria. The T is for thermos, heat, and AQ aquaticus. It's a bacteria that lives in, remember we talked about extremophiles, organisms that can live in very extreme conditions, hot temperatures, salty places. This is a bacteria that lives in the hot springs. And we've taken the polymerase from that bacteria now because it can live in hot places. It's more stable under high temperatures. And Taq polymerase now is going to be used in the thermocycler to polymerize the DNA. This process was invented by a guy named Kary Mullis, and he received the Nobel Prize for this technique. And basically it's a copy machine for copying DNA. Pretty useful tool. We're going to stop right there. There are a variety of other techniques to look at DNA and other kinds of technologies involved with DNA. We're going to talk about a few of them. But for now, if you have questions about anything in the slideshow, please bring them up in class and we'll discuss them. You should now be able to go to the Google Docs and take your little quiz. Be ready to do the DNA fingerprinting lab in class.